So hello everyone, welcome back to my another video. In today's video we'll be covering the glycolysis, which is considered as one of the most not one of the most hardest reactions in the in our syllabus, but one of the most hard to remember reactions. So today I'll be trying my best that I can make it easier for you to remember, because NEET is everything about remembering things, because you have really less time to solve a large amount of questions. So it is better if you remember some things than to understand it. So these are one of the things. So it is the part of the chapter named respiration in plants, from the 11th ncrt. You can check the ncrt. I'll be uh, teaching, I'm not teaching, I'm just explaining you guys. I'll be covering a bit more which is given in the ncrt. I'll be telling some of words which are not given in ncrt, but it has been taught by my teacher, so I'll be sharing it with you. So first of all, I would like to give the credit to my teacher, because these are not my this is not my data or not my notes. So if you like this, thanks to him. So let's start. So the first molecule in glycolysis is sucrose. Sucrose is a 6 carbon containing molecule. This is also known as sugar. Sucrose is divided into two molecules that are glucose, that is also 6 carbon containing molecule. Oh, wait a second. Sucrose is a 12 carbon containing molecule. Sucrose is a 12 carbon containing molecule. I'm really sorry. Sucrose is converted into, divided into glucose and fructose. Fructose is also 6 carbon containing molecule. So glucose is the one which then continues the reaction. That's why the reaction is called as glycolysis. Now the enzyme, now one of the most important things about this reaction, and every reaction covered in the respiration chapter, is are the enzymes. So I'll share some of the tricks that I use to remember the enzymes. So here sucrose is converted into glucose and fructose. So here the enzyme name is, wait, I just forgot. Wait. Here the enzyme name is, oh my god, okay. What is the enzyme's name? I always I always forgot the first enzyme name. I know every single enzyme name after this. What is the enzyme? Okay, let me check. Oh yeah. So then so the enzyme's name is invertase. Now I don't really have any trick for this enzyme, the first enzyme, but I can assure you I have every trick for all the next enzyme. So just wait, okay? So the first enzyme used here is invertase. This is embarrassing. Okay, now we continue the reaction from glucose. So glucose is now converted into glucose glucose 6 phosphate. Glucose 6 phosphate. Now here yeah. So here glucose 6 phosphate is formed after glucose. Glucose is converted into glucose 6 phosphate. Now to remember this enzyme used here, what here we have 6 phosphate. So the enzyme's name is hexokinase. So as we know that hex is used for 6, and hence the enzyme's name is hexokinase. So hexokinase is the first enzyme of the glycolysis. So glycolysis doesn't start from the sucrose. Glycolysis starts from the glucose. Okay, this is a lot of teachers. I'll not name teachers. I'll not blame teachers, or teachers are definitely more knowledgeable than me. But a lot of teachers doesn't state this, that glycolysis doesn't start with sucrose. It starts with glucose. So you have to count these steps from the glucose. When the glucose divides, when the glucose converts, from the glucose you have to count these steps. So this is the first step of glycolysis, not sucrose when converted into glucose and fructose. This is not the first step. The first step is when glucose converted into glucose 6 phosphate. Now glucose 6 phosphate is converted into fructose 6 phosphate. Now as you can see here in this step, the second step, there is nothing, there is not addition of anything, there is no addition of a phosphate group, not nor any oxidation, nor any reduction, or not any addition of hydrogen or oxygen. So here it is just an isomer of glucose 6 phosphate. So the compound, I mean the enzyme used here is isomerase. Isomerase. It's isomerase. So the enzyme used here is isomerase. Also one thing, when glucose converts into glucose 6 phosphate, ATP is used. ATP is utilized. So ATP is converted into ADP plus phosphate ion. And this phosphate ion then joins with glucose at the 6th position and forms glucose 6 phosphate. Now here's the trick. Every time when ATP is utilized, always the enzyme which is used in that reaction ends with kinase. Okay, ends with kinase, and the prefix of the enzyme is always the product formed. Now here it's not the product's name, here it's hexokinase, but you'll understand when we will move further. So here ATP is used and phosphate ion is involved, hence the enzyme's name end with kinase. Okay, you have to remember this. Okay, now glucose 6 phosphate is converted into fructose 6 phosphate. Here isomerization is done, and hence the enzyme's name is isomerase, and the product form is fructose 6 phosphate. Now this fructose 6 phosphate is converted into fructose 1,6 bisphosphate. I don't know if you have learned nomenclature yet, but when we name complex compound, we add bis, not bi, tri, di, like this. We add bis, tris, like tetra, case. Okay, so here one more phosphate ion is added to fructose 6 phosphate at the first position. As you can see, 1,6 bisphosphate. So here one phosphate ion add means here ATP is utilized. And what did I say to you? When ATP is utilized, ATP converts into ADP plus phosphate ion. I just said you that when ATP is utilized, we add kinase in the prefix of the enzyme. So here enzyme should end with kinase, right? Here enzyme should end with kinase. Now what about the suffix? What about the suffix of the enzyme? So the suffix of the enzyme should end with the product's first name. What is product's first name? Fructose. But so this enzyme name is actually phosphofructokinase. Why? Because phosphate ion is added, right? So the enzyme name is okay, I'll write it again. So the enzyme's name is phosphofructokinase. Now these are the first 3 steps of the glycolysis. These are the first 3 steps of the glycolysis. These are also known as the preparatory phase of glycolysis. This is also known, these 3 steps are also known as preparatory phase of glycolysis. Okay, also one thing you should know, that the first step of the glycolysis is irreversible. Okay, as well as the 10th step of glycolysis is also irreversible, the last step. Okay, now the fructose 1,6 bisphosphate is converted into 2 molecules. It is converted into 2 molecules that are, first is DHAP and second is PGAL. The full form of DHAP is dihydroxy acetone phosphate. Acetone phosphate. And the full form of PGAL is phosphoglyceraldehyde. Now this same situation we had when sucrose was converted into glucose and fructose, and we saw there that glucose converted this path there onwards, right? So here also one has to continue the path. So here the PGL is the one which continues the path. Now what about DHAP then? What happens to DHAP? So DHAP converts into PGL. How? By using the isomerase enzyme, which was used here as well. DHAP and PGL actually isomers of each other, and DHAP is converted into PGL because there is no use of DHAP if PGL is going to continue the reaction. So therefore DHAP is converted into PGL. So here DHAP is converted into PGL and enzyme uses isomerase. Now what about the enzyme which is used when fructose 1,6 bisphosphate is converted into these both? The enzyme's name is aldolase. Now this is one of those enzymes which you have to remember, like the invertase enzyme. I don't really have a trick for this one, but you should know when there is nothing happening with the substrate. These are called as substrates. ATP is specially known as substrate. So there is nothing in the substrate, then the enzyme's name you have to remember. Like I have also remembered, but yeah, this enzyme I didn't remember, so I always forgot this enzyme, so never mind. But I remember this enzyme name is aldolase. I also remembered this enzyme. I'll show you the trick. It's pretty BS trick, but okay. So as you can see here the word comes do means two, right? So here two products are formed, therefore aldolase. So I hope this makes sense. I mean, this doesn't make sense, but you can remember it that way. Okay, so as I said, that PGAL then converts continues the cycle. Now the point to be noted from from here onwards is every single compound that is going to form after this would be two molecules of those compounds, because DHAP is converted into PGL, so there are not two PGL. So further all the compounds after this are going to be two in numbers. Okay, so I'll be just multiplying this by 2. Okay, now PGL is converted into phosphoglyceraldehyde is converted into I was talking about that PGL is converted into 1,3 bisphosphoglycerate. 1,3 bisphosphoglycerate. As you can see, the phosphate ion is added, and when the phosphate ion is added, kinase comes into the picture. Kinase. The enzyme has to end with kinase. But here comes the exception, because here the enzyme doesn't end with kinase. Because why? I have my theory does imply here, because here ATP was utilized in both the reaction. Here in the third reaction and the first reaction, ATP was utilized. Here ATP is not utilized. Here ATP is not utilized. Here NADH2 is utilized. I mean, not utilized. NADH2 is formed. NADH2 is formed, not utilized. NADH2 is formed. How? I'll show you. So kinase will not come. Kinase will, although phosphorus is added, but kinase will not come in the prefix of the enzyme. So how the NADH is formed? Let's see that. NAD plus is converted into NADH plus H plus, and this will all be going in two numbers. I mean 2. Okay, and also two phosphate ions are also being obtained, which are given to the compound. Okay, now what about the enzymes thing? Now comes our next trick, that is related to NADH2. Now whenever you see NADH2 or whenever you see NAD in any of the reactions, I'm not just saying about glycolysis, any of the reactions of the respiration chapters, you have to use dehydrogenase in the prefix of the enzyme. Okay, prefix or suffix comes like suffix comes at the first, or prefix, I'm not really sure. If I'm doing it wrong, I'm really sorry. So okay, here dehydrogenase will come at the end. Okay, I'll just say at the end. Now what about the suffix? What will come first? Now, when NADH2 is used, always take the reactant's name. When ATP came, we use product's name. When ATP came, we use product's name. But when NADH2 is used, we take reactant's name. What is reactant's name? Phosphoglyceraldehyde. We'll remove a phospho. We'll take glyceraldehyde and dehydrogenase. So the enzyme's name would be glyceraldehyde dehydrogenase. Glyceraldehyde dehydrogenase. Okay. Now this 1,3-bis phosphoglycerate is converted into 3-phosphoglycerate. Now, as you can see here, one phosphate ion is removed. Okay. So here, So here in this case, the phosphate which is removed, it is used to form ATP. And what is the number of ATP formed? 2, because as all reactions are going in number 2. So I will just note it down. All of these molecules are 2 in numbers that we obtain. Okay. So the phosphate ion removed here is given to the ADP compound to convert it into ATP. So 2 ADP are converted into 2 ATP as they are given provided with phosphate ion. Now, wait a second. Yeah. So remember my first trick that I told you. Whenever ATP is involved in the substrate level, we use kinase at the end of the enzyme, as I said. And whenever ATP comes, we use the name of product. But in this case, we don't use the name of the product. Yeah. Wait a second. Yeah. So what I said. So yeah, we do use. Okay. So my theory is doesn't debunk. So what I said, that whenever ATP is used, we use kinase and we use the name of the product form. What is the product form? Phosphoglycerate. So what would be the enzyme's name? Phosphoglycerate kinase. Now the next thing formed is just an isomer of this compound, that is 2-phosphoglycerate. One thing I would like to say is wherever I am writing glycerate in NCERT, it is given as glyceric acid. So it would be better that if you remember it as glyceric acid, because my teacher has taught me as glycerate, so I remember that. But you should remember the way it is in the NCERT, okay? Because that's the way NTA would ask in the paper. So here as you can see, first the phosphate ion or the phosphate group was on the 3rd place, and now the phosphate group is on the 2nd place. So what happened here? It became an isomer. And what are we writing the enzyme's name from the start for isomers form? Isomerase. And also here isomerase, but this time we'll not use isomerase. This time the enzyme's name would be mutase. Okay. This is another enzyme that you have to remember. It doesn't relate anything with my trick. Okay. Now there are only 2 steps remaining in the glycolysis, and these 2 steps are filled with exceptions. The first step is, of course, 2-phosphoglycerate converts into 2-phosphoenol pyruvate. The full form of PEP is phosphoenol pyruvate. Now, to remember the enzyme of this compound, as we can see that phosphoglycerate, glycerate is converted into phosphoenol pyruvate. So there is no yeah, there is no addition of phosphate or any removal of hydrogen, anything like that. So the enzyme's name would be something else, not the way, or no, not dehydrogenation, not dehydrogenase or kinase would be added at the prefix. Now what would be the enzyme's name? The enzyme's name is I have a trick. The enzyme's name is enolase. Enolase. One thing here is that H2O is H2O is released. H2O is released. This is the only reaction, only reaction in the whole glycolysis that where H2O is released. That is the reason why I said that last two reactions are very different type of reactions, right? So here H2O is released and the enzyme name is enolase. Now how to remember this enzyme's name? What is formed? Phosphoenol pyruvate, enol. So here also enol. So enolase. I think it's easy. Now the last reaction of glycolysis is when 2-phosphoenol pyruvate converts into 2-pyruvate. I think you can spot something here. So if you are smart enough, or if you're not smart enough, like it doesn't matter, you can see that phosphoenol pyruvate, it was phospho. Now there is no phospho. Phosphoenol pyruvate. Where is the phospho? Enol. Let's forget enol for some time. Where is phospho? Phospho is gone. Phospho is gone. Which means here the phosphate is given to ATP. ADP I mean. And here ATP is found. Here ATP is found. Here ATP is found. So ADP is provided with phosphate ion, and here 2 ATP are formed. This reaction and this reaction, these are only reactions where ATP is found. And I said, and my trick applies. See, my trick is the most superior trick you can see on the YouTube. Okay, so I said that whenever ATP involves, we have to add kinase in the prefix of the enzyme. So we have to add kinase. And as I said, when ATP is formed, or whenever ATP is involved in the substrate level, we have to use the product's name for the suffix. So the enzyme's name would be pyruvate kinase. And by the way, these two main tricks are related to ATP and NADPH2. So the ATP trick is whenever ATP is involved, we have to add kinase at the end and the product's name at the start. And whenever NADH2 is involved, we have to add dehydrogenase at the end and the reactant's name at the start. Like here, NADH2 is involved, we add dehydrogenase at the end and glyceraldehyde, that is phosphoglyceraldehyde. So glyceraldehyde at the start. So the reactant names at the start and the dehydrogenase at the end. Okay. So 2-pyruvate is formed, hence the enzyme name is pyruvate kinase. I would like to mention one more thing, that wherever ATP is formed in the reaction, wait a second, yeah, whenever ATP is formed in the reaction, it is known as substrate level phosphorylation. ATP is formed. I'm saying ATP is formed, not ATP is involved. ATP is also involved in this reaction. But here ATP is utilized. Whenever ATP is formed, we say it as substrate level phosphorylation. From substrate level phosphorylation, we get 2 ATP. We get 4 ATPs. 2 here and 2 here. But also 2 ATP are utilized here. So net gain of ATP is 2 in the glycolysis. But you should not forget about NADH2. We are getting 2 NADH2 here. Okay, so for context, 1 NADH2 equals to 3 ATPs. So net gain of ATP would be, here 2 utilized, 4 obtained, 2 ATP directly we got from the glycolysis. Second, we get 2 NADH2, which when multiplied to 3, 6 ATP. So the net gain of ATP from the whole glycolysis is 8 ATPs. The net gain of whole ATPs from the glycolysis is 8 ATPs. I just explained you every single thing from the glycolysis. I also try to make it easy for you to remember. Also, don't forget, like me, this first enzyme. Okay, like no one asks about the first enzyme because glycolysis doesn't start from here. Glycolysis starts from glucose. So, and I just shared you every single trick for every single enzyme. Like for some enzymes like mutase and um, some enzymes are there which I don't have much tricks about. That is mutase, enolase. Now enolase I've given you trick, that phosphoenol pyruvate one. But some enzymes are there which I don't have a trick. Only one enzyme actually, mutase. Baki aldolase and enolase I've given you tricks. So this was the glycolysis. I'll be bringing on video on Krebs cycle and the ETS and also the link reaction. Link reaction is only one reaction, so it's very easy. I'll be bringing on videos on that as well. So yeah, let's meet on. Let's see. Thank you.